everyone, so today we will learn about ocular embryology. So let's start. For ocular embryology, only ectoderm and mesoderm take part in developing structure. Then neural growth form and it will invaginate and form neural folds. So here is the neural folds. Then neural folds will continue grow to each other and fuse, and there will be the formation of forebrain. Invagination in the area of forebrain from optic pit. Okay, optic pit is indentation formed in the inner surface of neural tube on the forebrain region. Approximately day 25, optic pit will form optic vesicle. During optic vesicle development, what happens is optic vesicle expand till mid-surface ectoderm, and then interaction between optic vesicle and a surface induce that ectoderm to thicken into lens placoid. So here is a lens placoid. Here, this one. And then this is lens pit and optic stalk. Okay, lens placoid invaginate lens pit, then form lens vesicle. At the same time of the lens vesicle formation, optic vesicle invaginate to form double layer optic cup. Optic visual development, what happen is developing optic vesicle and optic stalk have a groove on their inferior surface called optic fissure. Okay, this one is optic fissure, and then the fissure will fuse at week 7. Before the closure, mesenchyme enters the fissure and moves into the cavity of developing optic cup. Okay, the hyaloid artery carries nutrient to the developing lens until the production of aqueous humor occurs. And yeah, this is hyaloid artery and this is a hyaloid vein. Hyaloid artery will increase in number when lens matured and will remain as central retinal artery. Okay, this is the overview. We have optic cup, optic stalk, and lens vesicle. Move on with formation of other structures. First one is development of retina, and here is the information. Okay, this one is development of the lens. Also, we have development of iris and ciliary body. This is development of choroid and sclera. And this is the layer that form choroid and sclera. Okay, next is a development of vitreous. Lastly is a formation of eyelid and conjunctiva. Okay, this one is a very important, which is timeline of ocular embryology.
Hi everyone, now we're going to learn about vitreous humor. This is the structure of vitreous humor. Now the characteristic of vitreous humor is it is transparent, colorless, gelatinous or jelly-like structure, acellular, and it fills 4 feet of the cavity of the eyeball, which makes up 80% of globe volume. Next, for the structure, it composed of 99% of water, which is about 4 ml in volume, hyaluronic acid, and a network of randomly oriented collagen fibrils. Let's move on to the attachment. The attachment of the vitreous humor is mainly at the vitreous base, which is here. And there are also some additional attachments, which are macula, at the optic disc, and also at the back of crystalline lens by hyalodiocephalar ligament of Weger, or also known as Weger's ligament. Also, the strongest attachment of the vitreous humor is at the vitreous base. Now, the division of vitreous humor. Basically, vitreous humor is divided by two parts, which is the vitreous cortex and also vitreous body. Vitreous cortex lies adjacent to retina and ciliary body. Most important thing is it is tightly packed with collagen fibers, where the density of collagen fibers is greater in this peripheral part. So, the condensation of these fibers form a vast anatomic membrane, which is divided into anterior haloid membrane and posterior haloid membrane. For posterior haloid membrane, it remains loosely attached to the retinal limiting membrane. Let's move on to the vitreous body. Unlike vitreous cortex, vitreous body has a less dense fibrillar structure and it is a true biological gel. Other than that, it is more liquefied than outer zone, which is the vitreous cortex. It is also consists of Cloquet's canal, which is here, and Cloquet's canal running down the center of the vitreous body from optic disc to the posterior pole of the lens. For additional information, the circle area is the ora serrata, and anterior haloid membrane is located anterior to the ora serrata. And here is the Berger space, which is located between the posterior lens and anterior of the anterior haloid. Berger space is a structure that adherent in a circular manner by means of Weger's ligament. Now we have come to the functions of vitreous humor. The first function is to provide structural support to retina posteriorly and lens anteriorly. The second one is it acts as viscoelastic shock absorber. The collagen fibrils and hyaluronic acids will form gel with viscoelastic properties in the vitreous. Next, its function to maintain image clarity. The hyaluronic acid will form large open coils, and these coils will stabilize the structure of collagen fibrils, thus minimize the light scattering. Vitreous humor also stores metabolites for retina and lens. As you can see here, the metabolites for lens and retina are different. And lastly, it acts as gel barrier, where the gel properties will prevent solutes reaching the retina from the anterior segment. The last one is the age-related vitreous changes. The first one is light exposure. Too much exposure of light will cause oxidation and will increase cross-linking of collagen peptide chains. Hence, it will cause hyaluronic acid degradation. Next is the vacuolation, where gel will liquefy. The third one is vitreous degeneration will occur centrally. It occurs at the site where the collagen concentration is at the lowest. Next, the collagen fibril, which no longer separated by hyaluronic acid, will form hyaluronic acid clump, and this clump will become visible opacities. Last but not least is the firm attachment of vitreous loosens. So when the vitreous detaches completely, the peripapillary attachment becomes visible clinically, which also known as waist ring. So this is the example of vitreous problem. As you can see, there is a floaters in the vitreous, and there is also a vitreous detachment where the vitreous doesn't attach anymore to the retina. So that's all for this topic. Thank you for listening.
Hi everyone, in this video I will be explaining to you guys about the uvea. Uvea is basically comprised of three different structures, which are the iris, ciliary body and choroid. Uveal tract is highly vascularized and is the pigmented layer of the eyeball. From the picture, the one that I drew in pink are the structures of the uvea. We will be looking further into each of the structures later, but first let's take a look at the choroid. Choroid plays an important role as the main vascular supply to the outer retina, so it is a highly vascularized layer compared to the retina and sclera. Choroid is also thicker around the posterior pole than the anterior pole. Choroid contains four main layers, which are the Brooks membrane, also called as basal lamina, choriocapillaris, stroma and lamina fusca. Basal lamina is the innermost layer of choroid. It is a compound basement membrane between retina and choriocapillaris. Choriocapillaris have white ball capillaries that provide nourishment to the outer retina. Stroma is a pigmented layer that is composed of loose collagen, melanocytes with some elastic fibers, and a lot of blood vessels. Lastly, lamina fusca lies adjacent to the sclera and it is the outermost layer of the choroid. Lastly, let's take a look at the function of choroid. Firstly, it provides nutrients to the outer retina. The dark pigmented choroid absorbs excess light. And lastly, the suprachoroidal space provides pathway for the posterior vessels and nerves to pass through. Now that we have done with the choroid, let's take a look at the next structure, which is the iris. Iris is a thin circular structure located anterior to the lens. It also has a central aperture called the pupil. Iris can be divided into two regions by the collarate. The regions are the pupillary region and the ciliary region as shown in the picture. Iris is the thickest at collarate and thinnest at the root. The average diameter of the iris is 10 to 11 mm. Iris can be divided into four main layers, which are the anterior border, the iris stroma that contain the sphincter muscle, anterior epithelium that have the dilator muscle, and lastly the posterior epithelium. Next, the functions of pupil. Firstly, it provides channel for aqueous flow between the posterior and anterior chamber that is important in controlling the eye pressure. It broadens luminance range for vision. It controls optical aberrations. And lastly, it provides depth of focus during accommodation. The changes in the pupillary size depends on two factors, which are the light reflex and the near reflex. That's all about the iris. Next, we will discuss about the last structure of the uvea, which is the ciliary body. The composition of the ciliary body can be divided into three categories, which are the ciliary muscle, ciliary stroma, and ciliary epithelium. There are three different types of smooth muscle fiber in the ciliary muscle, which are the longitudinal, radial, and circular. The ciliary stroma is highly vascularized and contains loose connective tissue. Ciliary epithelium are the outer pigmented epithelium and the inner pigmented epithelium. The drawing shows the structures that make up the ciliary body, which are the trabecular meshwork, pars plana, pars plicata, longitudinal fibers, radial fibers, and circular fibers. The functions of the ciliary body are it involves in accommodation control by the ciliary muscle. It also produces aqueous humor. Let's go into details about the production of aqueous humor. The production of aqueous humor can be divided into three phases, which are the formation, composition, and also the drainage system of the aqueous humor. Firstly, the formation of the aqueous humor can be due to three different ways, which is firstly diffusion caused by the osmotic pressure gradient, ultrafiltration, and active secretion that accounts for 80 to 90% of aqueous humor produced. Next, the composition of the aqueous fluid. Aqueous fluid are free from protein, cells, and other large molecules. Ascorbic acids, amino acids, and glucose are actively transported to supply the cornea and lens. There are also high concentration of lactate due to anaerobic glycolysis that takes place in lens and cornea. Lastly, the drainage system of the aqueous humor has two different pathways. Firstly, trabecular meshwork road, and secondly, uveoscleral road. Let's take a look at the first pathway, which is the trabecular meshwork road. The aqueous humor flows into the trabecular meshwork through the Schlemm's canal, then into the collector channels, the aqueous veins, and lastly the episcleral veins. This route is sensitive to pressure. The increase in outflow related to the increase in the intraocular pressure. The second pathway in the drainage system is the uveoscleral route. The aqueous humor from anterior chamber angle move into the connective tissue spaces within the ciliary muscle, and then into the suprachoroidal space, and lastly into the sclera via the vortex vein. Lastly, the function of aqueous fluid. It maintains the intraocular pressure for optimal shape of ocular structures. It provides a low refractive index transparent medium for vision. And lastly, it delivers oxygen and nutrients, also responsible for removal of waste products from the retina and lens. We have come to the end of our discussion about the uvea. Let's take a look into the next chapter. Thank you for watching.
In this chapter, we're gonna cover about retinal layer. Retina composed of 10 layers, and we provide for you an acronym for easier to remember. In new generation, it only automatically examining patient's retina. By using this acronym, it will be easier for you to remember each retinal layer. In represent internal limiting membrane, new represent nerve fiber layer, generation represent ganglion cell layer, it represent inner plexiform layer, is represent inner nuclear layer, only represent outer plexiform layer, automatic represent outer nuclear layer, remaining represent external limiting membrane, patient represent photoreceptor, and retina represent RPE. In new generation, this is only automatic examining patient's retina. That's how you remember the retinal layer. Now we move on to cell in each layer. The outermost layer consists of RPE, contain retinal pigment epithelium to transport of nutrient metabolites to extra-retinal blood barrier. The next layer we have photoreceptor, consists of fluid which is responsible for peripheral vision and vision of low illumination, and cones for central vision and color vision. The next layer we have bipolar cell which relay information from photoreceptor to horizontal amarkin and ganglion cell. And in the same layer, we have horizontal cell that transfer information in horizontal direction and release inhibitor transmitter. The next cell, we have amarkin cell, which is a large cell body modulating information that reach ganglion cells. And the last layer, we have ganglion cell, which consists of 3 types. W cell, carrying information for pupillary response, Y cell for project lateral nucleus, X cell for response into visual discrimination. That's how you remember retinal layer and cell. Thank you.
Now I'm gonna talk about retina. So this is the cross-section of the eye, and there is the retina. Retina has 3 distinct regions. The first one is optic disc, second one is macula lutea, and the third one is peripheral retina. Now I'm gonna talk about optic disc. It's very pale pink in color with diameter 1.5 mm, and all the retinal layers terminate here. Next is macula lutea. It's located near the fovea. Macula lutea is 5.5 mm in diameter, and it also a dark area in the retina. Plus, it is the most sensitive part of the retina, and it also called as yellow spot or area centralis. Next part of the retina is the peripheral retina, and it is divided into 4 regions, which is near periphery, mid periphery, far periphery, and also ora serrata. Now, I'm gonna talk about retinal blood supply. For your information, all the artery enters retina through the optic disc. One of the artery is the choroidal capillary bed, which provide nutrition for outer retinal layers. Next is the central retinal artery, which provide nutrition for inner retinal layers. Next is the capillary network. The first one is the deep capillary network, which lies in the inner nuclear layer. The second one is the superficial capillary network, which lies in the nerve fiber layer and ganglia layer. Next is the cilio-retinal artery. This artery is the one of the vessel that enter the retina from the edge of the disc. Do you guys know where is the capillary free zone? Yes, it's only located at the fovea.